Hey everybody. Good morning or good afternoon or evening, depending on where you are in the world. Thank you for being here today. We're really excited to kick off the BPS MGO seminar series today. And if you haven't already, this is a reminder to please join the BPS MGO subgroup. And we'll be posting updates about speakers on the MGO Twitter. And yeah, so we're going to kick things off today. Our first speaker is Dr. Vijay Ramani, uh, who is a professor at UCSF and Gladstone Institutes. And he's going to be talking to you today about chromatin structure.
Thanks for that introduction, Camille. Thanks everyone for tuning in. For the next 20-ish minutes, I'm really gonna I'm really gonna talk about work that in part I always say this is the most important slide of my second. This is one of the two most important slides in my slide deck. Right? I'm going to talk to you about two stories today. I'm really going to touch on work from the person who just introduced me, who happens to be a graduate student co-advised between my lab and Ethan Ehrlicher's lab at UCSF. So I'll talk a little bit about Camille's work, which really focuses on trying to understand how the single molecular structure of chromatin fibers can actually potentiate their ability to form condensates at the mesoscale. And then I'll spend most of the talk talking about work that was really led by Marty Yang, who's a postdoc in my lab, with help from Sean Wong and several other folks in the group, on a new method that we've developed, really a computational method applied to a molecular method that we invented in my lab a few years ago that allows for us to now measure genome-wide at single molecule resolution what we're calling nucleosomal structural distortion. That may seem confusing now. Hopefully, it will be less confusing after the next 20 minutes. All right. So at work in my lab, I'm assuming this audience needs no introduction to chromatin writ large, but uh, I'd like to start us off by introducing a technology that really powers work in my group. And this is a technology, a molecular measurement technology that we invented about 4 years ago that combines highly active, non-specific adenine methyltransferase footprinting with a very particular type of DNA sequencing to map protein DNA interactions on individual intact chromatin fibers at very high resolution, at near nucleotide resolution. We use highly active methyltransferases. So the methylation reactions that we're doing should really methylate most accessible DNA in the context of chromatin. That's largely going to be the linker DNA in between nucleosomes. It's a it's a non-specific methyltransferase. So this isn't like MSSS1 or MCVIP1. This isn't a methyltransferase that methylates nucleotides in a very particular sequence context. This is a methyltransferase that should promiscuously methylate its substrate in most sequence contexts. And we think this is important for mapping protein DNA interactions at high resolution. And then the last bit is that it's an adenine methyltransferase. So as far as people have looked, there doesn't really seem to be much adenine methylation in mammalian epigenomes. And so this is an entirely orthogonal methyl mark that we're depositing to mark accessible chromatin that doesn't crosstalk with cytosine methylation. So it enables the, say, coincident detection of an endogenous epigenetic mark and then a mark that is telling us something different about chromatin structure. Okay, there's nothing new under the sun. And so when we developed this approach, we were heavily inspired by methyltransferase footprinting approaches that were really pioneered by groups like Steve Hennekoff, Peter Jones, Mike Clade, Dirk Schubler, Bas van Steensel, and many others. All right. So we can methylate chromatin and non-destructively mark where the protein DNA interactions are, but how do we read it out? And the second bit of innovation in our approach was that we leveraged advances in third generation single molecule long read sequencing to natively detect the adenine methyl marks that we were installing to mark accessible DNA. And so the way this works very briefly is that we throw this DNA, we purify the DNA that we've methylated, put it on a very particular type of sequencer. In our case, it's a single molecule PacBio sequencer. this similar approaches have also been developed for Oxford nanopore sequencing, but my lab basically exclusively uses PacBio. The way that the PacBio sequencer works is you drop the DNA into a very tiny hole, a so-called zero-mode waveguide that enables the detection of single molecules worth of fluorescence. The way that PacBio sequencing works, they call it single molecule real-time sequencing. And the reason they call it that is because you drop your DNA complexed with an evolved rolling circle polymerase that goes around in circles along a covalently closed circular DNA template, incorporating fluorophore conjugated nucleotides. And sequencing occurs when the zero mode waveguide is effectively used to detect incorporation events at single molecule resolution in real time. And that movie of successive incorporation events is decoded on the sequencer. It turns out that the sequencer is, this polymerase, I should say, is so sensitive to the methylation state and modification state of the bases that are being sequenced that it can reproducibly pause or stall in very particular ways when it encounters methyl marks. And I've kind of shown that in this cartoon here. And so we hypothesized that we would be able to decode where the methyl marks were on our footprinted DNA as a function of sequence by basically developing computational models to predict and classify these pausing events. And then this is something that postdoc Colin McNally in my lab did a few years ago, wrote the requisite machine learning models to actually carry out this classification task. I wouldn't really be talking here if this didn't work, and it turns out it works quite well. So this is data that we generated that really Noor Abdulhai and Colin generated with help from Geeta Narlikar's group now 6 years ago. Each line here is an individual molecule that we've sequenced. We've colored the regions that we think are methylated in purple and the regions that we think are unmethylated in this light blue. And I hope it's quite clear to you that there's some patterning here. I'll tell you that this is the patterning we expect. So what we did was we created chromatin fibers on template DNA comprised of 9 tandem repetitive copies of the Widom 601 nucleosome positioning sequence. And you could tell these are, in fact, the 9 tandem repetitive copies, followed actually by about 300 nucleotides of bases that should stochastically assemble nucleosomes. So this weird patterning here is actually to be expected. But we were able to compute what our error was for positioning the dyad and measuring linker lengths using this control data. And we could safely conclude that our approach really gives us near nucleotide resolution of where nucleosomes are on single molecules at very high throughput. This is just a few hundred molecules, but this scales to millions of molecules. And this can be done in vivo as well. And we've published, as I'll show you in a little bit, we've published a fair bit of work on this. This is for the citation for Noor's first paper from when she was a PhD student. But for the methods developers in the audience, you'll know that the most important part of any methods development task, sure, you need to show it works, but you also need to name the approach. And so we lovingly, this also works in vivo, but we lovingly refer to this approach as the single molecule adenine methylated oligonucleosome sequencing assay, or SIMOSA. And we were fortunate enough, we published this back in 2020, and we were fortunate to publish this at the time along several much larger groups than ours, including John Stam's group, Brad Bernstein, Will Greenleaf. In the last 4 years, we've been really excited by just how applicable, generalizable, and generalizable the SIMOSA approach has been. And so I like to say we've been kind of expanding the SIMOSA suite of technologies and continuing our close collaboration with Geeta's group. We published a story last year that had the good fortune of being the cover story of NSMB in last October, where we basically showed that we could use SIMOSA as a super powered biochemical assay to really resolve how, in this case, nucleosome sliding enzymes can actually restructure individual chromatin fibers. We've done the requisite technology development to actually miniaturize SIMOSA so that we can make these single molecule chromatin accessibility measurements in clinically relevant samples. This was published earlier this year. This is data from actually a patient-derived pediatric osteosarcoma line. And what we've done is we've looked at the nucleosome positioning patterns and protein DNA interaction patterns at single molecule resolution at highly occupied CTCF sites in this patient-derived cell line. And these happen to be fibers we profiled in vivo where CTCF was bound. You can see this beautiful CTCF footprint here. We see fibers where nucleosomes are occupying the CTCF site, and then also presumably these fibers where the accessibility has been made, but CTCF has come off, right? So we think it's a very rich source of information for really understanding how chromatin is structured in vivo and in clinically relevant samples. We have a paper coming out actually in 2 weeks. This is the amazing work of Megan Ostrowski, who's now a PhD student at UCSF, Alex Pollan's group, and Marty Yang, where we've added a time component to SAMOSA measurements. And now we do what we call replication-aware single molecule accessibility mapping, or RUSM, where we feed cells BRDU. We then train the requisite convolutional neural networks to actually decode whether BRDU is present in a given region of DNA and map the footprints. And so now we have a view of how the chromatin fiber is structured 10 to 15 minutes after fork progression. We're very excited about this new method we've developed. And then I'll show you a little bit of a slide from our latest work that was really spearheaded by Camille on the panel. And this is work, again, in collaboration with Ethan Arliker's group. This is available on the bioRxiv, I should say, where Camille has figured out how not only to reconstitute and map the single molecule structures of chromatin fibers, but also by combining SAMOSA with imaging, she can actually measure how individually structured chromatin fibers coalesce to form so-called, I won't say liquid-like, but so-called chromatin condensates. And I'll show you a couple of slides of what that looks like, where Camille has done single molecule mapping, where she can say create chromatin fibers with a very preset nucleosome density, that is a number of nucleosomes per unit length DNA. There's nothing to see in this imaging channel, but as soon as Camille ratchets up the nucleosome density to something that's getting to become more physiologically relevant, she starts to see these beautiful chromatin condensates with properties that she's really, really nicely characterized in this preprint. And then she can also map the underlying single molecule structures that constitute these condensates. I'll show one little piece of information that really Camille discovered that I think is really exciting, but we can extend these measurements to actually ask how complexes that mobilize nucleosomes might impact the ability of chromatin to condense and its other properties. And Camille noticed something really interesting when she added a co-activator, an ATP-dependent chromatin remodeling complex called RISC in the switch SNF family, to condensates. She could actually make the condensates dance. And we have plenty of speculation in our preprint of what might be happening here, but I just wanted to kind of leave you with this movie and just say that there's a lot we can do with these assays that enable single molecule measurement of chromatin structure. Okay, so I think I have like 9 minutes or so left. And so in the last 9 or 10 minutes, I'm really going to spend this talk telling you about our latest attempt to try to infer what I'll call, again, nucleosome structural heterogeneity from single molecule footprinting data. I don't really have the time to go into too much background on why we're interested in this topic, but I will say that we're drawing heavy inspiration from the pioneering work of the late Jonathan Whittam, but also work from Frank Pugh's group, Steve Hunikoff, and many others, including Vasily Staditsky. Really, the question I'm trying to answer is, can SAMOSA actually measure the internal structure of individual nucleosome core particles? And I'm hoping I can convince you that the answer to this question is yes by the end of this talk. So the beginning of this talk really starts with a technical details that I think will be interesting to this BPS audience. The way that we call nucleosome footprints from SAMOSA data is that we engineered a graphical model called the neural network hidden Markov model. For the initiated in the audience, it's really a neural network that feeds into a two-state hidden Markov model. Those two states those hidden states that we're trying to infer are accessibility and inaccessibility. What we're trying to solve is an inference problem where we're trying to infer the path of accessible and inaccessible states conditioned on the underlying sequence that we're sequencing and the probability that that methylation can be detected on that sequence if the sequence is accessible or inaccessible. It turns out that in the way that we've structured this graphical model, there's really only one hyperparameter. It's a bit serendipity that this happened. It's how Colin kind of coded up this model. But that hyperparameter really is, it can be described as the transition probability in the hidden Markov model component of the model, right? And what Marty in the lab realized just by playing around with the model was that if he tuned this transition probability, he could, and this is kind of obvious to those who've done any of this type of modeling, he could force the model to state switch, right? So by increasing this transition probability, he could force the model to effectively toggle more frequently between the accessible and inaccessible states. The interpretation of this, right, is taking the footprints that we've called and breaking them up into smaller, what I'll call for the rest of the talk as sub footprints. We're taking the same single molecule, we're Viterbi decoding that same single molecule 3 different times. We're only changing the transition probability in the HMM and thus breaking up these calls into smaller and smaller calls. Okay, so we can do this, and it's kind of obvious that one could do this. It would be obvious to anyone who's worked with hidden Markov models you could do this. Most people who've worked with hidden Markov models would just say what you're doing is you're populating less probable state spaces, and so you're just calling noise. So we needed to show or even test that possibility. The null model here is that we're basically calling noise. We're taking a perfectly good nucleosome caller and making it crappy. And so first, we could just visually show us breaking up these footprints into smaller subfootprints. This is what this data looks like graphically. We're taking the same 300,000 nucleosomes that you can see here from this length distribution of footprints, really nicely coincides with the expected amount of protected DNA for nucleosomes. And we're breaking those up into smaller and smaller footprints. So now we're just going to ask, do these subfootprints reflect biological structure? We're going to take these subfootprints and we're going to align them back to these parental reference footprints. And we figured that if this approach really was biologically relevant, we should see specific patterns. We maybe thought that if we calculated the accessibility at these different T's, we'd see something symmetrical, reflective of the pseudo-symmetric structure of the nucleosome core particle. But we also figured that the actual subunit structure of the nucleosome should fall out. So if we looked at the sizes of placed subfootprints with respect to the predicted nucleosomal dyad, the center of the nucleosome core particle, maybe we would see footprints reflective of H2A, H2B dimers and H3H4 tetramers. H3H4 tetramers, right? Maybe we would see footprints reflective of hexasomes comprised of a tetramer and a dimer, right? And maybe that hexasome might be accompanied by a dimer. Maybe it wouldn't, right? And in this case, it'd be a proper bona fide hexasome. And then we figured, as Jonathan Whittam would have predicted, maybe we would also see nucleosome core particles that display different extents of what he called breathing. That is the propensity of the edges of the nucleosome core particle to stochastically unspool from the nucleosome core particle. I hope this is making sense to folks. I'm moving quite quickly. But to make a long story short, it seems like this works. So the intranucleosome accessibility was patterned, but this is what we were most excited to see when we actually plotted the enrichment of these subfootprints with respect to the reference footprint midpoint. We actually saw this beautiful enrichment for intensities consistent with hexasomes, tetrasomes, dimers, but also this vertical line, which we think is suggestive of subfootprints that result from symmetrically increased accessibility at the edges. All right, so we think that what I'm showing you here is effectively like a Z-stack of like about 500,000 nucleosomes. Can we actually further dissect this data at single nucleosome resolution? And so we came up with an unbiased clustering approach that basically breaks that average down to 14 distinct structural, what we call types. And I'll just break down these types. These seem to be the most accessible clusters. We think these are consistent with highly breathable nucleosomes where we actually see evidence for a tetrazome, but we also see evidence of labile, what we think are dimers, but also what we think might be hexasomes, tetrasomes, and also rarer, these very short footprints that we think are consistent with dimers, but also TFs. We also see these kind of QR codes that look like this. we think these happen to be the least accessible nucleosome types, and we actually think these are consistent with chromatosomes, nucleosomes that are associated with linker histone H1. But then we also had these types, and we were kind of curious what the hell is going on here, right? We can kind of break these horizon plots down into what the accessibility is at the highest transition probability we computed. What we see is focal accessibility at very specific points. This is accessibility right at SHL2, which we thought was very interesting. We can also take just another cluster. This is accessibility right at SHL 3.5. I'll point out without going any further that this happens to be right where the join is between H2H2B and H3H4. And so we speculate that maybe what we're capturing are disassembly or assembly states or the coordinate action of transacting factors as they say dock on the nucleosome at very specific sites like SHL2. Okay, so in the last 2 minutes, I'll try to answer three very obvious questions that might have come up that you might have had during this talk. First, where are these nucleosomes genome-wide? We can map the enrichment of these different types across mappable and unmappable regions of the epigenome, and we see there's some logic here. So the least accessible nucleosome types seem to be enriched in heterochromatin. The most accessible nucleosome types seem to be enriched in euchromatin. Another obvious question, how are these represented at a TF binding site? Marty recomputed these distributions just at SOX2 OCT4 bound sites. These are going to be enhancers and promoters that are involved in the core pluripotency circuit in mouse embryonic stem cells where we did all these measurements. He can see that these breathable subnucleosomes are enriched at bona fide SOX2 OCT4 bound sites, and they're depleted at unbound motif matches that are just randomly strewn across the epigenome. He can compute how these patterns are actually statistically co-occurring on 2 kilobase fibers that encompass these entire enhancers. We're very excited by this. So these are patterns that are statistically enriched compared to background, where we see this breathable nucleosome, what seems to be a tetrazome, and then a dimer. So this is probably a hexasome, right? That we're actually footprinting distinctly at these SOX2 OCT4 bound enhancers. And then one final question, does the protein actually directly control the abundance of these nucleosomes? So we collaborated with Elzo de Wit's lab at NKI and used these small molecule sensitive degrons to basically erase SOX2 at the protein level in these cells, and then did these same experiments. And so what Marty could see, we were quite excited by this, is that if we get rid of SOX2 at the bona fide SOX2 OCT4 sites, we see an enrichment, we see an upregulation of these breathable subnucleosomes, and then perplexingly, we see loss of these inaccessible nucleosome types, consistent with actually SOX2 playing two roles, right? It's creating these very short footprints, but it's also somehow stabilizing wound nucleosomes in the vicinity of its binding site. And so we think this might have something to do with the so-called pioneer factor hypothesis that's been posited by Ken Zarrett and others. All right, so I've told you about Samosa. I'll leave this up and actually I'll just say this is the most important slide. This is our amazing lab, our amazing collaborators, and I'll just leave this up for questions. Thanks so much.
Thanks, Vijay. That was great. If anybody has questions, we're happy to take them now. Just yeah, Vlad.
Hi Vijay, nice, very nice. I'm wondering about this breathable nucleosomes, right? I mean, you mentioned that in your essays you are seeing some sort of symmetrical breathing, and it's kind of long thought that the breathing is kind of asymmetrical. So I'm wondering if the method that you're using now here, you can distinguish between symmetric and non-symmetric breathing.
It's a really good point, right? And I try to be careful with my semantics here, right? We see symmetric methylation, right? But a methyltransferase assay integrates over the dynamics, right? Over the time of methylation. And it's irreversible methylation, right? So if you have a nucleosome that's doing this, you're going to average out to this, right? So I think you need to pair this with like a really well-designed DSM-FRET experiment or something, right? To correlate the two, right? And so it's a really good point, and I should say that we observe symmetry, right? But that's probably the consequence of dynamic asymmetry. Yeah. I think it's interesting to think about what that means, right? I mean, the TF is also sampling for some amount of time, right? And so if the nucleosome is doing this and the TF is in the volume for that amount of time, like it's the same, right? So yeah.
Yeah, thanks.
I have a question. So to what extent is Samosa sensitive to like kind of the higher order structure of chromatin? Like if a nucleosome is in a very kind of densely packed region, do we get sort of a different readout than if it was sort of in an expanded region?
So we've shown in a few published papers that Samosa captures heterochromatin fiber structure. We've further shown that the fiber structure in heterochromatin is not quite what people thought it was by microcopal nuclease mapping. What it really we actually see that there are long NRLs in heterochromatin. They're just fiber that the representation of single fibers with long NRLs is lower than in euchromatin, right? Um, I think what one would like to do to answer your question is come up with methylation reagents coupled to like particles of different size, and then do footprinting reactions, right, to see how does the amount of methylation change as I add larger and larger things in, right? We think ECOG2 is larger than micrococcal nuclease and DNase-1, but it's clearly not so large that it is unable to methylate heterochromatin well. We don't think that higher order structure is it's going to have some effect, right? But not enough that like it's not like we're tremendously biased against footprinting mononucleosomes and heterochromatin. Does that answer your question?
Yeah. Thanks so much.
Yeah.
Hi, Vijay. I have a question actually about like one of the last slides. I don't know if it was the last one. Like the fact that depleting SOX2 actually also doesn't like makes this one. Yeah. So do you think it's a structural thing, like the binding to one side will make chromatin nearby closed, or do you think it's a recruiting thing? Because I can see also that happening, right? That SOX, like Vlad probably will mention it, but we know that many of these places are like SOX oct, so then you're gonna have oct flying around, being able to bind to other nucleosomes, right?
Yeah. Like how am I allowed to speculate? I'm gonna
yeah, sure thing.
Okay, yeah. So here's what I think, and this goes back to work that Patrick Kramer's group has published, but also a really nice preprint from Hayden Saunders in Geeta's lab. So HMG box proteins can interact with the nucleosome. Hayden has solved a structure with Yifan Cheng's group of HMGB1 bound to a nucleosome, and it turns out it binds very nicely at SHL2. Patrick Kramer's group published a Nature paper several years ago of SOX2 bound to a nucleosome, and surprise, surprise, it's also bound at SHL2. And I think that structure, it's difficult to reconcile with Nico Thoma and Dirk Schubler's work, which shows SOX2 nicely bound at the entry-exit point, right, of the nucleosome. I haven't shown it here, but we actually see the same thing. So what I think we're seeing is multimodal, motif-dependent versus independent interactions. So when you have a SOX2 OCT4 regulated enhancer, right, you're going to have factors like switch SNP creating accessibility rather, and you're also going to have SOX2 OCT4 binding its motif, right? The preferred position that is motif encoding is almost certainly the protein bound at its motif and a nucleosome at the positioned such translationally positioned such that the entry exit is right abutting the motif. That doesn't rule out the possibility that there are motif independent interactions where SOX2 might be stabilizing a nucleosome via its motif independent binding mode, where it's going to, at high enough concentrations within a given volume, interact with the nucleosome at SHL2. And so I suspect that's what we're seeing. We don't really have the data to say that, so it's speculation right now.
Okay, thanks so much. We should probably move on. But thank you, Vijay.
Yeah, thank you, Vijay. And I'm actually, this led, I think, pretty nicely to what I suspect Vlad will be talking us about. We're gonna be now talking about the same thing with an even higher resolution, if that's possible. I think Vlad uses this concept I really like of the computational nanoscope. Just a bit of a background. Vlad, I know Vlad because he was my PhD supervisor back in Münster in Germany. He recently moved to Babes Bolia, I guess I pronounced that more or less similarly. And yes, and so excited to hear what he has to tell us about. And Vlad, the floor is yours.
Thanks, Sean. Yeah, it's great to be back in the MGO community. And thanks a lot for the introduction. Well, so I recently moved to Babes Bolyai University in Cluj. It has and I hope that with this we start to put Romanian universities on the map of this kind of research that we are doing. So for those of you that are not aware of the methods we are using, we are mainly using molecular dynamic simulations to study how transcription factors interact with nucleosomes. And just to give you a very brief introduction, basically this is just you take an atomistic resolution structure, atomic resolution structure. You can of course use other representations, but we stick to the atomic resolution structure. You have the positions and the velocities in the beginning, and then what you basically do, you just generate a movie in time of your molecule. Do you see the mouse, actually?
Yes, we do.
Okay. So you basically generate a movie in time of your molecule. So in reality, you just watch the molecule moving. And you are limited here by the time step that you can use, and this is usually between 1 femtosecond to 4 femtoseconds these days. And you do this by integrating Newton's equation of motion, and you know that the force is also the minus derivative of potential energy. And from this, you can calculate the forces, and using an algorithm, I'm not going to go into details, you generate next positions in time. You can use different software to do that. And to give you a short kind of overview of what we can do these days, it's basically, if you look at biologically relevant transitions, microsecond, millisecond, seconds, now you can think is that we basically simulate about 40, 50 nanoseconds a day when you use CPUs only, and about 50, 100 nanoseconds a day when you use the GPUs. Now, the main approximation is this equation here, is the force field, the potential energy function, and very briefly, all these like bond lengths, angles between 3 bound atoms, and so on and so forth, electrostatic van der Waals interactions, electrostatic interactions, they all are the so-called force field parameters that would give you the accuracy of the simulations. Now, luckily, we have pretty accurate force fields. Now, we use this kind of system where we solvate the molecules in a box of water. This is replicated infinitely in space, so it's a periodic system, basically. This has some theoretical background, but don't have time to go into details. And you have the salt, the ions around your system. You can simulate NPT or NVT thermodynamic ensembles. So you can do these physics-based simulations. And we are very much interested in transcription factors because they can do all these great things. Basically, you can transform skin cells into all kinds of different cells by just inducing a few transcription factors. And I think that's absolutely fascinating. And one of the transitions that we are very much interested in is basically this reprogramming to pluripotency. So basically, you take a somatic cell, fully differentiated cell, and you can revert it to a so-called pluripotent cell, which has the capacity to differentiate in any type of cell in your organism. And you can do that by just 3 transcription factors, sometimes even 2 are enough. And we know that OX4 and SOX2, that Vijay also mentioned before, are very much at the core of this transformation. Now, we also have the pioneer transcription factor hypothesis that Vijay also mentioned, coming from Ken Zaret and others, which basically say that pioneer transcription factors are those that are able to bind, let's say, closed chromatin states. Basically, DNA were occupied by nucleosomes. And the question is now here, what is it happening when these transcription factors are binding? So basically, you have this kind of silent state or this competent state where the transcription factor binds and creates a little bit of motion around here. And then you have all the recruitment of other factors that can lead to this active state. Now, all good, but we know that chromatin looks more or less like this, and like very much these pictures. And all these nucleosomes, the units of chromatin, are very tightly packed into chromatin fibers. And we know actually that they are not as tightly packed as it used to be thought. So we have here a bit of sizes for these chromatin fibers. So the question is, how do these transcription factors actually recognize their DNA binding sites in this particular environment? Now we look mainly at OCT4 and SOX2. OCT4 pluripotency factors, OCT4 binds to the major groove of the DNA with 2 DNA binding domains. The linker between the 2 domains is also a very interesting story that I'm not going to touch about today. SOX2 binds to minor groove, bends the DNA very strongly. And there were the 2012 and 2015 papers from Abdel Soufi and Ken Zeret, which basically showed very briefly that OCT4 and SOX2 and also KLF4, which is the third pluripotency factor used for reprogramming, they all bind to this kind of closed states of chromatin. Some naive interpretation of the structures at that point about how this can be. But nevertheless, we basically embarked in this adventure to try to understand if we can use molecular dynamic simulations to actually understand how OCT4 and then SOX2 bind to nucleosomes, but also what are the effects that this binding produces on nucleosome dynamics. And you all know these pictures, I guess, those of you who are present here. It's a core nucleosome with the whole histone tails here that play a very crucial role. And that's one of the reasons we really like the simulations, is because we are able to actually look at these nucleosomes with the tails attached to them, which is very nice. Of course, the tails pose different problems for the simulations, but nevertheless, if you look at the structures of these nucleosomes, most of them, if not almost all of them, are without these tails. Now, if you look at the dynamic nucleosome, this is more or less the pictures you get that when you look at. These are the most flexible parts. You see the tails, the histone tails moving everywhere. The histone core, pretty rigid. I only show one snapshot here of the histone core, but it's pretty rigid. And you have these motions in the linker DNA that you see quite clear here. Now, we reviewed a couple of years ago the motions that you can actually extract from such simulations. And this would be this kind of breathing motions that Vijay also mentioned, right? So this kind of detaching of the ends of the nucleosome from the nucleosome core particle. But you can also look at the twisting motion in the nucleosomes. And sometimes you can also look at these intergyre motions, although for these classical simulations in which you don't apply any kind of force are a bit tricky because of the time scales that you are sampling. These motions are pretty happen on very long time scales. So when we look at breathing from the nucleosomes, we introduce this kind of order parameters, which we call these two angles, gamma 1 and gamma 2. Basically, just these two projections here that you see on this picture of these vectors along the linker DNA and the coordinate system. And this gives you these histograms like this, where you can see open conformations will be populated here, closed conformations will be here. So you can characterize this motion in a relatively simple fashion. Shan actually did a lot of simulations of the genomic nucleosomes. We are also interested in genomic nucleosomes because we took basically nucleosomes that are bound by OCT4. We know they are bound by OCT4, this one and this one. And we look at the motion. And surprisingly, even with the histone tails attached in the system, we are able to observe this arrow, this black arrow point you to some pretty open conformation of the nucleosomes. So we were able to observe breeding of the nucleosomes even when the histone tails were there. Now, unsurprisingly, when you remove the histone tails, the nucleosomes go mad, and you have much more breeding. That's not surprising, has been observed by others. But actually, what was interesting to see is that we still kept the sequence-dependent pattern, even when the tails were removed. So that was kind of interesting that not only the tails are responsible for keeping the nucleosome closed, but also the DNA sequence plays a role in this scenario. So now we did all this work, mainly Shan did actually the work with free nucleosomes. We tried to understand how they behave. And then we asked ourselves, okay, so what happens when pioneer transcription factors encounter chromatin fibers? And then you see here a picture more or less updated with this less compact structure of the chromatin. And then the question is, how do these factors actually act on the dynamics of these fibers? So now if you look at the timeline of the pioneer factor concept you see that it started all the way in late 80s. And you see here in blue all the genomic experimental data. So most of these data came from genomic studies, basically. Now you see the review of Kenzeret and Carroll that basically established the concept of pioneer transcription factors. And then you see the first biochemical study, which is in light blue here. And then on the right, you see when the structures of these pioneer factors with the nucleosomes in complex started to be resolved. And these are in red here. Now, the first structure was the SOX structure coming from Patrick Kramer's lab that Vijay also mentioned. And then we had other structures of other pioneer factors. But actually, the first computational study was a cross-grain simulation by Tan and Takada, which have also looked at this kind of motions induced by OCT4 and SOX2 binding, mainly by SOX2 binding. And actually, our work, our main publication describing how OCT4 actually opens nucleosomes was actually the first atomistic simulation of this kind of motion. And then you see all other structural that started to pile up in the recent years. To make a long story short, and we saw this already published work, where basically we looked at OCT4 binding to two different nucleosomes. On one of them, we saw that OCT4 is kind of stabilizing a partially open conformation of the nucleosome. You see there is no sampling of the closed nucleosome in the absence of OCT4. In the presence of OCT4, sorry. And this is a binding on this edge of the nucleosome. So this was one of the things. But one of the very exciting things that we saw was basically this massive opening of the nucleosome when OCT4 was bound in this particular one simulation. And you see here a snapshot here on the left of this. So basically we asked ourselves, okay, what happens here? Is it something that happens only once, or is it something that we can actually reproduce? And now we reach the limits of molecular dynamic simulations, especially when you have the histone tails attached to the nucleosomes. But we still were able to use some different methods. I don't want to go into details too much, and show that basically the patterns that we see, it's still when OCT4 is present, using different methods now here. I'm not going to go into details of what kind of methods. We just applied some biasing potentials here. And we saw that in the presence of OCT4, you still have the more opening of the nucleosome. Now, the most exciting thing, and that we are now trying to wrap up in a new manuscript, basically, was that when we depleted the tails, the histone tails, we saw opening in the presence and in the absence of OCT4, but the opening was still significantly larger in the absence of OCT4. We now extended the simulations, and I'm very excited to see the newest results coming from them, to see to what extent we can pinpoint what is the precise mechanism by which OCT4 impacts nucleosome dynamics, because in the simulations with the histone tails, we also have the histone tails, which have a a known role in basically nucleosome breeding. So we come up with this idea that OCT4 with these two domains, basically, one is bound sequence specifically, the other one is unspecifically sampling the nucleosome, and when it reaches this kind of position, it stabilizes this massively open nucleosome conformation. And we propose this model. Again, this is all published. But now what is interesting is, after our work, different structures of OCT4 nucleosome complexes came out. And the first one was actually this one on the right. And this showed nucleosome sliding of this, one of the nucleosomes that we simulated. Now, in this particular structure, OCT4 is bound to a piece of linker DNA, actually. And it was proposed that actually the nucleosome is slid completely comparing to the free nucleosome. So the proposal was that OCT4 somehow induces this kind of sliding, which was also pretty interesting. But in this case, you have OCT4 bound to actually free DNA. Now you have next paper, which was very interesting as well. Different OCT4 binding to different binding sites on kind of the same nucleosomes that we simulated. And again, you see this OCT4-induced opening, especially here. So lots of the concepts that we actually proposed in the simulations are now revisited by the experimental structures. But of course, there are also significant differences between the structures. And if you are curious about the differences and between the structures and our simulations, I can, of course, answer those questions. Now, recently, we shifted our interest towards other transcription factors. And here is a summary of all kinds of nucleosome conformations that you see in the experimental structures of pioneer factor nucleosome complexes, starting with this massively open nucleosome in the SOX11 bound nucleosome, with all kinds of different degrees of open nucleosomes depending on where the transcription factor is bound. So we came back to the paper of Patrick Kramer, in which they resolved the structure of SOX bound to two different sites on the nucleosome, SHL +2 and -2. And this shows a massive opening of the structure of the nucleosome in the structure. So we asked ourselves if we can actually simulate that. Can we simulate the opening? And first, we needed to create atomistic models starting from this structure. This is a the structure has a poor quality in terms of side chains, orientations, and so on. So we needed to build the closed nucleosomes from it, which was also resolved by Dodonova et al. But then you have also open nucleosome. You have to extend the DNA. You see in the structure the DNA is missing, so we had to actually extend the DNA to create this open nucleosome. And then you have to add the histone tails and create a model with SOX2 bound to a closed nucleosome, but also to an open nucleosome, because the question is, can SOX2 induce the opening by binding to the closed nucleosome, or is just stabilizing a partially open conformation? So we did all this, and we ran a lot of simulations, and these are now ongoing. It's ongoing work, and also we recently got a grant for this work, so I'm happy that this will continue. But to make a long story short, we again start observing this kind of opening of the nucleosome. You see this DNA radius of gyration, and you see that at least in one of the simulations, you do have this kind of opening. Not in all the simulations. Again, we reach the limits of what simulations can show us. But using our angle representations with these histograms, you again see the sampling of an open nucleosome started from a completely closed nucleosome bound by SOX2 in these both binding sites. Now, you may wonder how real is that, and I'm happy to answer that later. And basically, this is, again, relatively large opening that we observe by SOX2 binding to the -2 SHL site. We did not yet observe an opening of the nucleosome on the +2 site, which is interesting because it was kind of thought that the major opening comes also from the +2 site. So that's something that we are now currently investigating. But again, the whole opening of the nucleosome comes with the rearrangement of the histone tails. So we again come to the kind of same mechanism that we saw with OCT4, but now with a transcription factor that binds completely different. It binds the minor groove, it bends the DNA. So although the two transcription factors are very different, in terms of topology, in terms of DNA binding properties, it seems like the way they open nucleosomes is not so different. And with this, I would like to stop here. I think I'm sorry, I didn't actually timing. I forgot to switch the timing. So basically, it comes to the conclusion that, well, first of all, simulations are able to predict transcription factor-induced nucleosome opening, and I'm really happy about that because it was not clear that we can actually do that. I have to also say that we did a lot of experiments because at the time when we started this work, there was no experimental data about where the binding sites are and so on. So I didn't mention anything in that, but it's in our papers. All the experiments are there. So we validated every single binding site and so on and so forth. So simulations have predicted value in terms of predicting how transcription factors engage with chromatin, with nucleosomes and open nucleosomes. And also that the opening mechanism seems to somehow be similar when you look at very different transcription factors, which is interesting because in principle, SOX2 deforms DNA much more than OCT4, but still looks like, at least very preliminary now, that the opening mechanism is not much related to this kind of local DNA distortion. And with this, I would like to thank very much a lot of people actually. So most of the work, OCT4 work, was done by Shan. All the experiments that I didn't mention here, but they were absolutely crucial for all this work, were done by Caitlin. Then other people in the lab in Münster contributed a lot of Hans Schöller, which gave us all the support that we actually needed for this work. Then the SOX2 work was started by Daphne and is now continued in my lab in Cluj. Alexander also very supportive in at the time when I spent in Utrecht. All these collaborators, some funding from here. And just to give you a last slide about a city that you might have not heard of. This is common. This is Cluj. This is the university that I'm working at. It's probably one of the most important and most historic universities in Romania. This is the University Square, basically a very, very beautiful place. This is a view of the main square of the old city in Cluj. And this is basically the new institute that was established here and where our office that is actually extremely nice. And this is the bike road that you have to the institute with all these beautiful views over the city. And with this, I would like to thank you and welcome any questions, comments.
So thank you, Vlad. And Camille, you have a question? You want to go ahead?
Yeah. Thanks, Vlad, for a great talk. I was just wondering if, given the abundance of linker histone H1, if you'd ever tried to model a chromatosome and if so, what the time scale of breathing was like for the intact chromatosome.
Okay, so we have done some work on the chromatosomes way back, some years back. And we did see, at that time, there was still the debate about binding on diet, off diet. I think that's still not completely solved. We mostly worked on a model that proposed an off diet binding, but we haven't done any simulations that basically link transcription factor binding with linker histone. And I know there are experiments out there suggesting a potential competition between different transcription factors binding to regions where linker histone would bind. And I think the two recent structures of OCT4 bound to nucleosomes, they also propose something like that based on experimental data. But we haven't done yet any simulations in which we have the linker histone in the system. It's a bit complicated because the linker histone has also this huge C-terminal domain, right, which is a massive problem for simulations because it's not small. It's a very intrinsically disordered regions and it's huge. So, but yeah, it's one of the things that is definitely worth looking at.
Yeah, Vijay, you also have a question?
Yeah. Vlad, really nice talk.
Thanks.
I'm wondering, about so there's I think plenty of experimental evidence to suggest that switch sniff remodelers, for instance, are genetically essential for creating the motif accessibility in the first place, right? And then there's a little bit more evidence. There's some nice in vivo mapping work from Matthew Gerard's group that just got published. There is work from Krumizaka, right, suggesting that there are remodelers that can create these uh, non-canonical chromatin structures. So like overlapping dinucleosomes, where a hexasome has been pushed onto an octosome, right? Or two hexasomes have been pushed together, or two octosomes have been pushed together. Have you thought about modeling those? And then modeling TF interactions with those non-canonical structures, which almost certainly are getting generated at these regulatory elements.
You know how many things I would like to model? Yeah, sure. So these are great systems. These are amazing systems that you. The only problem that we are facing with atomistic simulations, of course, now you can do that coarse grain much easier probably. With atomistic simulations, of course, we face the issue of system size and computational resources for them. So that's why it's kind of tedious. But of course, I mean that would be, I think, the next step of simulating the effect of not only transcription factors but also other proteins have on nucleosome dynamics would be to actually consider this kind of arrangements of multinucleosomes, right? Where you can have not only dinucleosomes, but you can also have hexasomes in the system and all these different structures. And I think that would be fantastic, especially if we can start looking at genomic sequences, because that's where the trick is. So the genomic sequences are not very easy to handle experimentally. You probably know very well that, better than me. And that's where all these dynamic structures come actually probably much more relevant than in the typical Widom-like scenario, right? So yeah, I would love to model those. I would really love to model those. For now, we still do mononucleosomes, but yeah, I have written a couple of grants with dinucleosomes, unfortunately, and tetranucleosomes actually also, unfortunately not funded yet. So we got now funded the nucleosome story, but we are still far away from simulating the full transition, opening, closing, right? We still have, so that's the next challenge there, is how to get the simulations to really simulate a full opening, closing, because this we cannot do at the moment. This opening is very rarely happening, right? So we always have to trick it somehow.
Right. Yeah, thanks. Yeah.
Yeah, so there are no further questions. Thanks once again, Vlad and Vijay, for joining us, and everyone for being here. And I have to remind you once more, if you haven't, please sign up for the MGO subgroup of the BPS, and follow us on Twitter. I think the Twitter is MGO _BPS, and we're gonna announce the next webinars. So see you all around.
Thanks a lot.
Thanks.
Bye.